Fibrodysplasia ossificans progressiva is a rare disease that only occurs in 1 in every 2 million people. This genetic disorder causes the muscles and tendons of a person to calcify and become bone over time. This can be induced with injury and invasive surgery and grows progressively worse over time. It is caused by a genetic mutation on the long arm of chromosome 2 within the ACVR1 gene. This gene codes for the activin receptor protein, which is part of the bone morphogenetic protein family. It is part of the BMP signal transduction pathway using serine kinases, as the protein plays an important role in both the development and repair of our skeletal system.
The ACVR1 gene is 139,417 base pairs long. 3 different transcripts are created from the gene through alternative splicing. Each transcript results from a coding sequence of 1,530 nucleotides and creates a protein that is 509 amino acids long. While the ACVR1 gene has multiple variants, they all code for the same protein, with little to no difference in phenotypic effects, and each one can acquire this mutation.
When this genetic mutation occurs, a histidine replaces an arginine at the position of codon 206. This leads to a conformational change within the activin receptor protein that causes it to not bind as tightly to the inhibitory molecules. This causes the SMAD pathway to be activated at a higher rate, and thus the BMP pathway is fired more frequently. This causes soft tissue to turn into bone by changing endothelial cells to mesenchymal stem cells, which later turn into bone, thus calcifying the muscles and tendons within a patient.
There is no cure for this disease. In addition, like previously mentioned, any type of surgery is nearly impossible for the risk of increasing the damage to the muscles and tendons. Over time, it becomes progressively more difficult to do basic tasks like putting on clothes, walking, and brushing your hair. Many people with FOP eventually die from breathing complications because their lungs can no longer expand to get enough oxygen.
The most famous case of fibrodysplasia ossificans progressiva is that of Harry Eastlack, who lived from 1933 to 1973. The condition began to develop when he was 10 years old, and by the time he died, his entire body had calcified and he could only move his lips. To study the ACVR1 gene further, we amplified the small open reading frame of 3,060 base pairs and copied it into an E. coli plasmid. After finding the largest open reading frame, we located the forward and reverse primers. In order to copy into the E. coli plasmid, we tagged the primers with restriction enzyme cut sites that would allow it to fit into the plasmid's genome at the specific cut site of the MAL-E gene. To further extend the analysis of the ACVR1 gene, one would run a PCR with the new plasmid incorporated with the gene to amplify and make multiple copies. Having numerous copies then enables the gene to be modified in many different ways, and mutations can be added to see the effect that they have on the functionality of the gene.
To confirm that the gene was amplified correctly, one would run a gel electrophoresis containing the PCR protein products with negative and positive controls. Within a simulation, we saw that our protein was 99.65 kilodaltons, which hopefully would be confirmed with an actual experimental approach. With studying the ACVR1 gene, we can only hope to find a cure for fibrodysplasia ossificans progressiva in the future.
Good job.